All right, hey guys, today we're going to talk about structure and function of proteins. And so we're going to dive into protein topic 1.5, explain how changes in polymer subunits lead to changes in structure or function of a macromolecule. So by the end of today, you'll be able to explain how changes in protein subunits can lead to changes in structure and function. And so this is going to tie into an activity we'll do later on in class based on protein folding. All right. So discuss, what do you think would happen to an organism with a mutated DNA polymerase protein? So DNA polymerase helps synthesize new DNA. Synthesize means to create new DNA. So what do you think if there's a mutation in your DNA? What do you think is going to happen to the protein? All right, and so think about it, and we'll cover as we discuss these ideas. All right, so let's get into protein structure. As you can see that there's 4 levels of protein structure: primary, secondary, tertiary, and quaternary. And so each level builds on the others. So it's basically like building blocks. They build on top of each other. And so each level is held together by different bonds and interactions formed between amino acids. And we'll talk about the different bonds between the amino acids and between other structures for each different level. All right. And so proteins have directionality, so they're not symmetrical. So it's kind of like reading a sentence. The dog bit the man is different than the man bit the dog, right? So we need to first remember that you start with the N terminus, and this is the start. And it's the first amino acid. So this is the start. It's the beginning of the protein chain, and it's represented by NH2. All right, this is the N terminus. It's called an amine group. And so now we also have our C terminus over here. So remember, this is our start, and then this is our end. Again, the N terminus has your first amino acid, and your C terminus is going to have your last amino acid. And so the carboxyl group is free. So carboxyl is represented by COOH. And so all proteins have a primary, secondary, and tertiary structure, but only some proteins will form into quaternary structures. The directionality is important because proteins are built, like I mentioned, from N terminus to C terminus. So this matches how DNA is replicated from prime to 3' end. And so eventually, if there's a change to any of these, then it's going to affect the way the protein folds and functions. All right. So before we move on, let's talk about amino acids. So there are 20 amino acids. Each amino acid has the same peptide backbone. So it has the same amine group, carboxyl group. It has a hydrogen. And the only difference is that the R side chain is going to be different. And that's going to determine what type of amino acid it is. All right. So again, you have your amine group, which is NH2 or NH3 depending. You have your central carbon. You have your hydrogen, and you have your carboxyl group. The R chain is going to be the only thing different between amino acids, and it's what's going to determine the amino acid's property, right? Like I mentioned, amine group, it could be represented by NH2 or NH3. It contains nitrogen, so you can see the nitrogen right here. It's capable of hydrogen bonding, and the amine groups found in the R side chain will usually be positive charged. So this will usually be positively charged. All right, we have our hydrogen present, and this is because The hydrogen is required for carbon's 4 bonds. So because there's carbon, it needs a 4th bond, so there's a hydrogen. We have our carboxyl group, which is right here. It could be COOH or COO negative. And so this is called a carboxylic acid. It's also capable of hydrogen bonding. And the carboxyl found in the R chain will be usually be negatively charged. So this side is always positively charged, and this side is always negatively charged. Remember, this is the start, and this is the end. All right, and so this is the central structure of an amino acid. And what makes the amino acid differently, because remember we have 20 amino acids, is the R chain. So the R side chain varies between the 20 amino acids, and we're going to get into these in a little bit. All right, again, this is a summary. We have our amine, our carboxyl end, our alpha carbon, so our central carbon with the hydrogen attached, and the R group. That's the only thing different. All right. And so let's get into hydrophilic and hydrophobic. So we're going to be using these words throughout. Hydrophilic means that it likes being near water. Usually these are polar covalent compounds. So for example, salts and ions. And hydrophobic doesn't like being near water and are non-polar. So for example, oils and fats. So remember, anything polar, like water, is going to be with water. Non-polar, it's going to be hydrophobic. So remember, like dissolve like, so oil and water don't mix well because oil is hydrophobic and water is hydrophilic. So they wouldn't be able to mix. All right, let's get into hydrocarbons. And so functional groups are like molecular personality traits. And so hydrocarbons contain carbon and hydrogen. It's hydrophobic and it's stable. So there's very little interaction between these molecules. And these are all examples of hydrocarbons. You can see that it contains both a carbon and a hydrogen throughout. All right, so let's get into functional groups. So in AP Bio, we're going to have to focus a lot on functional groups. And so basically you have a hydroxyl, a carboxyl, an amino, and a phosphate. And so the parts of organic molecules that are involved in chemical reactions are called the functional groups. So if you substitute other atoms or groups for hydrogen, and so it gives organic molecules distinctive properties. And so this is what's going to make the hydrocarbons hydrophilic. It increases solubility in water, so it's able to dissolve in water because it'll become hydrophilic. And so examples are propane and propanol. Propane is a gas and propanol is a liquid. So adding OH into the hydroxyl is what changes everything. All right. And this is based on the functional groups. All right. And here you can see that even in hormones, females, when you add an OH, it's completely different than testosterone. All right. So the different structure affects the function. Here are more examples of functional groups. So you could see that you have your hydroxyl, your carboxyl right here, your amino, and then you have your phosphate over here. All right, let's get into ATP's importance. So ATP, it's our cell's energy currency. And so notice how there's phosphate groups. And that's where the energy is stored. And so when those phosphate groups are broken down, then those bonds are breaking down, then you are creating energy that the cell can use. And this is due to hydrolysis. Remember, hydro meaning you need water to go into the reaction, and lysis means it's going to slice, it's going to break these bonds in order to release energy. All right, so let's get into some questions. Do you think this molecule is soluble in water? Yes or no? And let's remember that we have these functional groups that make things hydrophilic. So it makes things be able to dissolve in water. These functional groups right here by adding OH, a hydroxyl. And so the answer here is yes, because you can see that there's OH. And so because of these hydroxyl groups, it means that it's polar and it's hydrophilic. All right. So remember that anything that's hydrophilic is polar, or anything that's polar is hydrophilic. So it is going to be able to dissolve in water. And so what does the term amino acid signify about the structure of such a molecule? So what do you think the term amino acid means? All right, and we know that amino acids, all amino acids have an N terminus, so the amino group. It also has a C terminus, which is the carboxyl group, and so that's what we call an amino acid. Remember that they also have an arch side chain that's different from one another based on those 20 amino acids. All right, we have another complex scenario. So which of the following statements best describes how organisms such as rabbits attain the carbon necessary for building biological molecules? So do you think that rabbits eat plants and use energy absorbed from the plants to make carbon atoms from electrons, protons, and neutrons? Do you think that rabbits eat plants and break down plant molecules to obtain carbon and other atoms that they rearrange into new carbon-containing molecules? Or do you think that rabbits eat plants and use water absorbed from the plants to hydrolyze carbon dioxide, which the rabbits breathe in from the air and use as a carbon source? Or do you think it's the rabbits eat plants and make carbon-containing molecules by causing carbon atoms that the plants absorb from the soil and stored in the cells of their leaves? All right, so you could see here that they're eating carbohydrates, and then they form this long structure. So what do you think this long structure is? All right, and so here you can see that there is some sort of rearrangement of the carbons. And so the answer here is B. So rabbits do break down plant molecules to obtain carbon and other atoms and rearrange them into new carbon-containing molecules. It can't be A because it's suggesting that you're making carbon from subatomic particles, and so that's nuclear fusion. That's not in biology. It can't be C because it talks about hydrolyzing carbon dioxide, and so this basically is photosynthesis, and rabbits can't do photosynthesis. Remember, only producers can, which are like plants. And then option D is suggesting that plants get carbon from the soil, but we know plants get carbon from carbon dioxide in the air during photosynthesis. So plants are able to make their own energy during photosynthesis, but we can't make energy, right? That's why we eat things like plants and veggies and things like that, to get this energy by breaking down these bonds and rearranging them to an energy form we can use. All right? And so remember we talk about R groups again. And R groups are chemical properties, have chemical properties and bonding abilities of the amino acid side chains, and are determined by the elemental composition. So anything that is hydrophobic, you want to look for a carbon and a hydrogen. So it has long hydrocarbon chains. And it has a big hexamer ring. So it has 6 sides. You can see the big hexamer ring right here. And so examples would include fatty acids, steroids, and anything with a benzene ring as well. Hydrophilic, remember that you're going to look for oxygen, nitrogen, or even sulfur. And so there's no charges on something that's hydrophilic. And so examples would be hydroxyl, carbonyl, and sulfhydride. Okay, so anything acidic would be hydrophilic as well. And so you're going to be looking for carboxyl groups and negatively charged. Anything that's basic is also hydrophilic, but you'd be looking for amine groups instead, and it has positive charges. All right. So remember that polar molecules are hydrophilic, nonpolar are hydrophobic, And acidic and basic substances are also hydrophilic, except that acids are negatively charged and bases are positively charged. All right, and again, like I mentioned, nonpolar are hydrophobic and polar are hydrophilic. And so these are the amino acids that are nonpolar and hydrophobic. And then these are the amino acids that are polar and hydrophilic due to the R group right here. So you want to look at the R group to determine whether or not it's nonpolar, polar, whether it's hydrophobic, hydrophilic. All right. So hydrophobic would have those hydrocarbon chains. So you look over here and you see that it has those chains, those rings. It only has carbon and hydrogen, so you can see that it has carbon and hydrogen. Over here, you're looking for oxygen or nitrogen with no charges. And so you're, or sulfur as well. And so here, like for example, you see sulfur, here you see nitrogen. And so in order to determine the amino acids properties, then you want to look at the R groups, okay? Which we said are different for the 20 amino acids. I know that some people do memorize like just all the amino acids and then memorize whether they're hydrophobic or hydrophilic. All right, but we're gonna practice based on these rules that we've been discussing. So look at the side chain in each box and determine if it's hydrophobic, hydrophilic, acidic, or basic. Please also determine if a side chain is capable of hydrogen bonding. All right. And so basically here, what do you notice here? And then hopefully you said you saw hydroxyl group right here. And the hydroxyl group, remember that that's going to make something be hydrophilic. So yes, it can hydrogen bond. All right, over here, what do you notice over here? I notice a benzene ring with an OH, a hydroxyl as well. And so the ring is hydrophobic, but because of this OH, it makes it capable of hydrogen bonding. So this is actually hydrophobic, but it can hydrogen bond. All right. So it's hydrophobic because of the benzene ring, but because of the OH, it can hydrogen bond. All right, what about this one right here, bottom left? And so remember that because of this negative charge, it means it's acidic, so it's hydrophilic. And so yes, it can hydrogen bond. Remember, anything that's hydrophilic can hydrogen bond. All right, and over here, so you notice how it has that positive charge. So this is basic and it's also hydrophilic. So it also can hydrogen bond. So all of these can hydrogen bond, except this one is hydrophobic overall. And the rest are hydrophilic. All right, so amino acids can exist in different forms depending on the pH. So usually when it's at a physiological pH, they're either ionized. And so here you could see it being ionized. It has both a positive and negative charge. Remember, that's what ions mean. And so the amino acid has both positive and negative charges. Here it's non-ionized and it doesn't have any charges. And so this is due to the pH. The way that you join proteins together, so how do you make proteins, is by joining amino acids together, sorry. And so notice how here water is leaving, so as a byproduct, so it's dehydration, and you're combining two things, so it's dehydration synthesis. And so the bond between these two groups right here is called a peptide bond. All right. So for your I'll go over the summary in a little bit, but this is making a primary structure, and that's why you have a peptide bond. And so you're joining two amino acids. Again, remember, you always need an enzyme to catalyze the reaction and speed it up. All right. And so now you're going to make a polypeptide bond. And so this is the secondary structure. And so this is where the protein starts getting its shape. So it could either be an alpha helix or a beta pleated sheet. And I'll show you a picture in a little bit. But this secondary structure is held together by hydrogen bonds between the backbone structure. All right. Again, you see water leaving as a byproduct. So this is dehydration synthesis. And you know it's synthesis because it's making it bigger. All right. Again, we need enzymes. And there's a hydrogen bond between the backbone structure. All right. So this is the primary structure. The first example we went over here. And so this is primary protein structure of a sequence of amino acids formed via protein synthesis. So the amino acid sequence is what determines how the protein folds at different levels. And so changing the amino acid sequence will usually cause the protein to misfold and stop working. We'll get into an activity, and so you'll be able to visualize this in class. And so this can lead to cells or disease. So an example would be sickle cell anemia. So again, we said that, like, for example, changing amino acids can cause there to be a disease or death. And so this is an example. This is a very extreme example, but in sickle cell anemia, there's only one change in the amino acid. Instead of a glutamic acid, which is hydrophilic, you have a valine, which is hydrophobic. And so that's going to change the folding, because remember, hydrophobic things like to be away from the aqueous solution. So it's going to fold differently than if it were to be hydrophilic and it'd want to be near that aqueous solution. And so this is what causes sickle cell anemia, and it causes that sickle red blood cell. So again, remember that the formation of a primary protein structure forms through dehydration synthesis, as we went over in those little animations. And so you need help from a ribosomal enzyme. And so the peptide bond specifically forms between the carboxyl group of one amino acid and the amino group of the next. So the peptide bond is covalent, and it's formed between the two amino acids, as we had seen in the other picture. So here you can see the peptide bond joining these two together in dehydration synthesis. And we said that you need an enzyme. And so the peptide bond is forming between the carboxyl group of the first amino acid and the amine group. So this is the amine group, and this is the carboxyl group of the first amino acid. And so it's forming it between this carboxyl of the first and this amine group of the second. So between here. All right, secondary structure is where the protein starts getting its shape. So like we mentioned, there's alpha helices and beta helices. And so it's held together by hydrogen bonding between the carboxyl and amine groups on the peptide backbone. And so there are no R groups involved. So you see that there's no R groups involved. And so looking at it close up, you can see the red boxes. Those are hydrogen bonds right here. And so the protein is following the rules of chemistry. So it's minimizing energy by forming these stable structures. And so notice how the peptide backbone is outlined in black. And the R group sticks out to the sides. So there's no R groups involved. And again, the hydrogen bonds right here are between the backbones, right? Between. And so tertiary structures is where proteins become functional. And so this occurs as the protein finishes folding. So it's controlled by interactions between the R side chains. So this is where amino acid properties we learned become crucial. Because hydrophobic collapse is away from the aqueous solution, and it's the major driving force behind the formation of tertiary structure. So here you can see anything that's hydrophobic is going to be away from the aqueous solution, and anything that's it's hydrophilic and it's near the aqueous solution, and hydrophobic is away from the aqueous solution. You also see that things that are basic and acidic, because they have those opposite charges, they like to be near each other. And so we'll practice protein folding in class. All right, and so hydrophobic collapse. So the driving force between tertiary structure is hydrophobic collapse. So here because something that's hydrophobic wants to be away from water and into the interior of the tertiary structure of the protein, you can imagine how this is what drives the folding. So anything black is hydrophobic, and like I said, it wants to be away from that aqueous solution. And so the white circles are hydrophilic and they want to be near the aqueous solution. All right, so let's get into the 4 types of interactions that stabilize these tertiary structures. And so one of them being the hydrogen bonds between the side chains. And so you could see the hydrogen bonds right here between the side chains. And so basically the hydrogen bonds are weak but numerous. Again, they're between the side chains and with electronegative atoms. So remember those first videos where we mentioned fluorine, oxygen, and nitrogen are the most electronegative atoms. And so there's a charge attraction between acidic and basic structures. All right. And hydrogen bonds are usually represented with these little like dots. So if you ever see something like that, just know it's a hydrogen bond. And so hydrogen bonds are very sensitive to changes in pH and temperature, and will be broken if the protein is outside of the normal pH or temperature range. And we'll get a little more into that once we start talking about enzymes. And hopefully we do an activity with that. The next bond we want to talk about that stabilizes to the tertiary structure are disulfide bridges. And so these are strong covalent bonds between cysteine and amino acids. And so, for example, between sulfur atoms present in the side chain and between 2 cysteine amino acids. And so these bridges are very strong and not sensitive to changes in pH or temperature. So the more disulfide bridges, it means it's a stronger structure because we said it's super strong and can't be broken down by changes in pH and temperature. And so the cysteine amino acid do not have to be near each other. All right. So here you see a disulfide bridge. And the amino acids, the cysteines, do not have to be near each other in the sequence to form the bond. So usually found in proteins exposed to harsh conditions. So either a high temperature or extreme pH, because nothing's going to happen to them, right? All right, so let's get into quaternary structure. So quaternary structure, let's just sum this up. So notice how hydrogen bonds and ionic interactions are sensitive to pH and temperature, but disulfide bridges, the more there is, the stronger they are. So the more stable that protein is going to be. All right. And so then we can move on to quaternary structure. It occurs when multiple proteins come together to form a protein complex. You can see all of this together. Their multiple proteins came together. All right. And so these proteins are all folded at the tertiary level. And so not all proteins are involved in quaternary structures, but they're held together by interactions between the variable side chains and hydrogen bonding between the peptide backbones. So for a quaternary structure, what's holding them together, the bond, is anything really. So it could either be a hydrogen bond, a covalent bond, ionic bond. But it's between the R groups of the different polypeptides, all right? All right, so an example here would be hemoglobin. Hemoglobin is a quaternary structure. And so it's your classic structure that you'd be seeing probably in your AP test. And so each color represents a different protein subunit that's already folded to its tertiary level. Another example would be looking at that sickle cell connection again. So the quaternary structures changes when the individual subunits are altered. And so because there's a mutation right here in the sickle cell, and then the proteins will stick very abnormally to each other, forming fibers that distort the red blood cell shape, giving it that like crescent moon shape. And so to sum it up, we have our 4 levels of protein structure: our primary structure, our secondary structure, we could either be an alpha or a beta. So alpha, it's like a ribbon, and beta goes pleated. We have our tertiary structure right here and our quaternary structure right here. Again, our primary structure is made up of amino acids, and it has peptide bonds holding these amino acids. Our secondary structure has hydrogen bonds between the backbones. Our tertiary structure, it's any bond, so hydrogen covalent ionic, but it's going to be between the R groups. And our quaternary structure, it's any bond, so hydrogen covalent ionic, but it's between the R groups of different polypeptides. So again, this is between the R groups of 1, and then this is between the R groups of many polypeptides. All right. So this is one polypeptide chain, and this could be 2 or more polypeptide chains. All right, and we talked about the denaturation. So we basically said that pH, temperature, things like that can affect the way proteins fold. And this is what we call denaturation, right? So denaturation is what happens when proteins lose their shape. And so they denature. So it's kind of like if you have origami and it unfolds into a flat sheet. So things that can affect this are environmental factors such as high temperature, high or low pH, salt, so high salt concentration, extreme pH levels. And so this is because it's disrupting those hydrogen bonds, those ionic bonds, and those disulfide bonds. So some proteins can sometimes return to their functional shape after denaturation, but many cannot. So the function is going to be affected because the structure is affected. Remember, structure affects function. Last but not least, we have chaperones, which guide proteins into folding. And so these provide shelter for folding polypeptides, and it keeps the new protein segregated from cytoplasmic influences. So you can see the guide, and it's providing shelter for the polypeptides. So this is the little shelter. It's the polypeptides are going in, and it's keeping the protein segregated. All right. And so here we have our little summary. You have your amine group, which is your start, your carboxyl, which is your end. Amine groups are usually basic, which means it has a higher pH. Carboxyl are acidic, so it has a lower pH than 7. You see that you have your hydrogen in the primary carbon, your carboxyl, your amine group, and your amino acid. The only thing that's going to be changing is the R group, and it's good to know the polarity so you know how it's going to fold. And based on the R group, you want to know if it's hydrophilic, hydrophobic, or if it's charged. So is it negatively charged or positively charged? Remember, if it's negatively charged and positively charged, it's going to be folding to the exterior. So just like a hydrophilic R group. And if it's hydrophobic, it's going to fold into its interior away from the aqueous solution. And here are the summary of the bonds between the primary, the bonds that are holding a secondary structure, bonds that are holding tertiary, and bonds that are holding quaternary. So pause if you need to, but I went over this already. And so basically, in summary, you want to remember that anything that's polar is hydrophilic, and so it's going to be on folding onto the exterior. Anything that's non-polar is going to be hydrophobic, and it's going to be folding towards the interior structure. All right, and then I'll go make another video going over these questions by the end of today. All right, thank you.